Okay, let's go. Welcome everybody to this H3D Foundation webinar series. We're thrilled to have Prof. Fidele Ntie-Kang, who's an associate professor at the Department of Chemistry at the University of Buea in Cameroon. I know Fidele very well because we work together on the Grand Challenges African Drug Discovery Accelerator. So we're so thrilled to have him here. And I'm going to begin by reading his biography so we have some context. Fidele Ntie-Kang is an associate professor of pharmaceutical chemistry in the Department of Chemistry, University of Buea in Cameroon, where he also heads up the Center for Drug Discovery. He completed a PhD in computer-aided drug design at the University of Douala in Cameroon, then moved to Germany, where he carried out postdoctoral research under Prof. Wolfgang Sippl, leading to a habilitation in pharmaceutical chemistry. He focuses on chemoinformatics for natural products research and is passionate about drug discovery from natural products identified from African medicinal plants. His major contribution is the development of the African Natural Products Database, currently containing more than 11,000 compounds and 1,700 species from all regions of the continent. He is also the solo editor of a book series on Chemoinformatics of Natural Products. He holds several awards, including the Calestous Juma Science Leadership Award from the Gates Foundation and the 2025 Royal Society of Chemistry Natural Product Reports Emerging Investigator Lectureship. He is also, as mentioned, a member of the operational leadership team of the Grand Challenges African Drug Discovery Accelerator. He's here today to chart a path towards the efficient discovery of natural products from African sources and to discuss how this can inspire the discovery of small molecules, including pseudo-natural products. So Fidele, thank you so much for being here. You're an incredibly busy man, so thank you so much for carving out the time for us. And to everybody, as usual, there'll be a Q&A at the end of the session, so please write your questions in the chat or note them down somehow. And I look forward to facilitating that at the end. So Fidele, over to you. Thank you so much.
Thank you very much, Nicola. Actually, I am really excited to participate in this webinar series. I would say that I've actually been following some of the webinars, and I'm quite excited to be the one to speak today. And my talk is going to be, as you introduced, natural products from Africa to provide or inspire solutions to global health. And I'll be talking by starting to narrate a story and try to imagine yourself in a context where you are sitting in a library in an African university, and you have access to all the digital copies of the master's thesis and the PhD thesis that have been published in natural product chemistry. At the same time, you also have access to some of the journal articles to which the university is subscribed. And you start checking through, and you're flipping through the thesis, and you want to see all the medicinal plants that were studied or investigated in that university. Now, by the time you look through this medicinal plants, you're going to see that each of them is well identified, belongs to a family, a place of harvest was recorded. In some cases, actually, the place where the sample has been kept of the plant and the reference number of the sample. And this could also be as well as animals and fungi and so on that have natural products in them. You want to see, okay, the traditional use of the species that was investigated. Then you go further to see the compounds that were found in there. You want to know if these compounds were active. You want to know which compound classes this belong to. You want to know what could be the potential applications of the compounds in drug discovery. Now, by the time you are done with all that information in one library, you would now think about the next library which you are going to visit. By the time you go through every single library on the continent of Africa, then you have a very huge repository of data. And then you realize quickly that a lot of this data is not accessible to the average African, and it's not also accessible to the large scientific community. So this is the wealth of knowledge that we started mining I would say almost 2 decades ago. And when we started mining this wealth of data, our interest was in the molecules, where our interest was in the species and the references in the literature. And then after that, we started looking also at the chemical structure files, and also beyond just the chemical structure files, because most of the cases we didn't have access to the samples of the compounds. Then we started looking at other external databases like PubChem, where you can have information about vendors of compounds. We started looking at other databases like NCBI, the taxonomy database, where you can have information about identification of the species, because we quickly realized that classifications of species often evolves over time. And so what is described sometimes in the literature as name of the species is different from one another as time goes on. So it was important to associate every species with a particular identification. And then we started also looking at other places where we can have more information, like looking at PubMed, where we can have information about the literature, information that is sometimes very scarce in the library, sometimes just a thesis, and you need to have all this information. So by the time we put all this together, then we started generating structure files that would be useful for drug discovery purposes, for studying interaction with proteins, for understanding the scaffolds, diversities, and so on and so forth, for predicting activities and toxicities. Now, at the end of it all, we realized if you put all this information in the web and everybody has access, then you have just built a gold mine, which can be both useful for African scientists as well as scientists from out of Africa. And so our efforts, that was where we started. Our efforts have actually led to what we call today the University of Douala Drug Discovery Center, which you see in the picture was the inauguration of the center in February last year, sometime around this time. So we are just like inaugurated for 1 year, but actually we have been existing for about 4 years. And we have so far 4 running laboratories, I would say. We have a natural product laboratory. We have a synthesis laboratory, chemical synthesis laboratory. We have a computer-aided drug design laboratory. And we also have a bioassay laboratory. So in essence, we have 4 laboratories running. And our vision has been to become a state-of-the-art drug discovery center in Central Africa, which makes use of natural products across the continent to identify drugs which are suitable for resource-limited settings. I will insist on resource-limited settings because drug discovery in Africa will require a scientist coming from abroad to undergo another training to be able to carry out drug discovery in Africa, because the realities on the terrain are quite challenging, and you will have to adapt to a lot of things. And of course, we want to serve our mission to serve as a platform for the catalyst and discovery of new drugs in Africa, exclusively led by Africans. Now, in my talk, I would talk about, I will show how the biodiversity of Africa could be both a challenge and an opportunity. I would also talk briefly about natural products and how natural products could inspire the discovery of new drugs. And I would, of course, focus on the African Natural Product Database, and I would show some case studies. Among the case studies I would show would be one of the recent works we just completed, which was the discovery of an HIV-1 latency reversing scaffold. And of course, we are going to call for collaboration, and we are going to have some conclusions. Now, I said before that the challenge has been enormous in the sense that to get access to data was the issue. And now that we are really having access to the data, we now ask what are some of the opportunities we have on the continent. Now, the first thing we'll say is that Africa is not a country, as many people will say. Africa is very diverse. We are not just diverse as a people. We are not just diverse in our genetics. Our environment in which we live is also diverse. You see, you will range from tropical forests to subtropical forests and savannas, to mangroves, to marine coastlines, to desert areas, and so on and so forth. So you see, Africa, that is already very huge. So every species in this whole continent tries to adapt to the environment where they are, and in the course of adaptation, they are bound to make metabolites, which they both need to survive in the environment and to defend themselves from invaders. Now, it so happens that a species is often interested in surviving in the environment, but in surviving, they have to make chemistry. Now, when this species makes chemistry, this chemistry actually there are primary metabolites and secondary metabolites, and then typically we'll call the secondary metabolites natural products, and it ends up that these natural products have biological activity. So in essence, we drive ourselves from biology to chemistry to pharmacy. But what has been happening in the past in Africa, and what is actually happening in most cases, is that there is always the need to do a kind of bioassay guided fractionation of species that are known to be used traditionally to treat particular diseases. So the traditional approach would be that if I am interested in finding anti-malaria compounds, then I would go to talk to traditional healers who have preparations for the treatment of fevers and so on. And of course, when I know this, with the help of a botanist, I can identify the species that is being mentioned in the traditional preparation. Then I would have to prepare extracts and try to identify which extracts are active, and then fractionate and further fractionate to get subfractions, which I can now hopefully be able to identify compounds in there. And if I succeed to identify the compounds and characterize them fully with the use of advanced techniques like NMR and mass spectrometry, then eventually I can now see if these compounds have the activity that is described in the botanical knowledge. And if that happens, then I say that I have validated the botanical knowledge with scientific information. But often, the cut-off activities of these compounds that are isolated are often very, very, very weak. So the explanation that often comes is that, oh, the extract acts synergistically, and so as a result of that, when you separate the compounds and you have purified compounds, then they lose their activity. But our own approach is, when we have collected all the data, we have the diverse compounds isolated from different species, and we have all of them, like now we have several thousands of them, then what we do is we want to carry out simulation. And in the course of carrying out a simulation, we are going to interact these compounds or their fragments with the drug targets, and the compounds that interact the best with their drug targets are the ones that we are going to suggest now for assays. In some cases, we actually assay the compound in itself, or sometimes we combine the fragments and assay and make new compounds, which are synthetic compounds, which means the compounds themselves are synthetic, but their fractions are obtained from nature. So we call them pseudo-natural compounds. And these ones are often also tested in the assay. So that's our own approach. Or sometimes we simply carry out a visual screen where we just screen the entire library, and then we now try to see what are the modifications we can carry out on the natural compounds to improve its activity, or to remove the expected or the tested toxicity that is seen in the compound. I would say Africa as a continent, it looks like we are diverse, but our problems are the same. One of the big challenges we have in Africa in drug discovery, particularly in natural products, is access to instrumentation. I would say, for example, an NMR, like the one you see in the picture, if you carve out North Africa and carve out South Africa, most likely you would have 2 or 3 functional NMRs on the continent. And this can be really challenging for African scientists who have to isolate the compounds and ship them out there and hope that some nice good Samaritan will be able to run their spectra and give the spectra so that they can be able to identify the compound in the sample. Now, there are many reasons why we needed to put all this data together, and I'll run through 6 of them. One of them was that the compounds in the libraries were in hard copies that were not digitally accessible from a distance. So in essence, that would simply mean that if I must study a species that was published in Kenya, I would either fly to Kenya, or I'll get someone get a hard copy, borrow a hard copy, and send it to me by DHL, which I hopefully will get after about 2 weeks, then send it back, which of course was not practical. So that's the first challenge. The second challenge is that most Africans themselves publish in journals for which the universities don't get subscriptions. So which simply means that the African scientist publishes their work, and the other African scientist doesn't have access to the work because the university is not subscribed. Then, of course, the herbarium infrastructure is quickly dilapidating. I remember just going to the field to identify a species, and to confirm it in the herbarium took almost like a week or so to travel there, and then you find the infrastructure. Actually, so nothing was digitalized, so you had to go through piles and piles of preserved plant material to be able to identify and be sure that that's the species we're talking about. And of course, because of this inaccessibility to that kind of information, several persons will be working on the same plant at the same time without even knowing. So there's duplication of efforts, and of course reproduction of the same results. Then of course, as the population in Africa grows, then people encroach into the forest and they want to construct on forest land or preserved land, eventually have access to the species and destroy the species, and many species become extinct over time. Then, of course, I will not dive into marine species because that's another story altogether for another day. Then we talk about the passing away of the older generation who are knowledgeable about information on traditional medicine, and eventually this information is not captured. So it becomes so important to capture this information and store it electronically so that future generations can have access to that. Now, a summary of all the efforts and the challenges that we faced in the course of our work has been published in this review. And I would say that recently also, we narrated a story of some of the challenges that Africans as a whole face in in health-related research, in not just drug discovery, and that range from several topics that we work together as CJ Fellows, and this was published here. Actually, we here provide some kind of roadmap because the long version of the paper is not published fully, but here we give a summary of a comment that just narrates some of the steps that could be taken to face some of the challenges, to remove some of the roadblocks that African scientists face in the course of trying to do research on the continent. Now, why are we interested in natural products? I would say quickly, the reason why we are interested are twofold. First of all, that Africa itself is biodiverse, so that alone, trying to investigate what is found in each of these species and how do these species survive, how they survive over the generations itself is quite a motivation. Another thing we say is that because many of the species have been used traditionally, we want to know what's in there. Is it that what we are consuming as products from these traditional preparations, are they healing us or are they killing us? And beside that, we know that with the use of computer-aided drug design technologies, we can actually fast-track the cost of identifying bioactive compounds from a collection of collection of compounds. So making use of this library will be quite interesting for us because that's going to be one of the ways of accelerating the identification of lead compounds. Now, if you compare natural products generally with synthetic compounds, we'll say that the main challenge will be that natural compounds usually come in very minute quantities because they come in complex mixtures, and the challenges of separating them from these complex mixtures is there. And then the challenge of identifying them too, because their structures are generally more complex than synthetic compounds. And so because when you are synthesizing compound, you know exactly what you want to obtain. And whereas if you are isolating compound, you don't know what you expect. So in essence, the challenge of determining the structure is quite more complicated. Of course, they have weaker bioavailabilities in terms of DMPK profiles, but because they are quite complex and they have many centers, it's quite intriguing to know how complex natural compounds are. And I always challenge people to say, there's no chemist who could sit in the lab and just imagine the structure of Taxol and synthesize it just without nature giving us the inspiration of that structure. So I would say the same thing for artemisinin and quinine and so on. Now, but in the market, you will see quickly, because of some of the challenges I mentioned, that only about 6% of compounds commercialized are natural products unaltered. Whereas if you go through the percentages, you will see that also 73% of small molecule antibacterials are natural products. And beyond that, about 50% of anti-cancer drugs are either natural products themselves or natural product mimics. So these statistics are there to tell us that natural products is not something of the past. But what is nature inspired is the question we have been asking ourselves. In trying to ask ourselves the question, what is nature inspired? I will go back again to my story of the African Natural Product Database. Now, we have currently collected about 11,000 compounds. And so what do we do with them? Because these 11,000 compounds come from almost over 1,700 species, which are plants, animals, bacteria, fungi. And we have been able to catalog all of this, and recently it was published in Nucleic Acid Research. But beyond just the compounds in the database and the species, which are information already available, we have also characterized the structural classification of the compounds in terms of their scaffolds and the diversities. We have been able to also give values, numbers of properties that are tied to ADMET properties, I mean absorption, distribution, metabolism, excretion, and toxicity. So which means that some of these numbers can quickly help for someone who wants to look for a compound which has some future in terms of from a drug discovery perspective. Beyond that, we also have all the information about the biological activities that were tested already. And beyond just the information about the biological activity, we also have NMR data and mass spec data, which can help you if you find a compound and you want to think that this compound is the one that you have in a database. Of course, you can compare the NMR with the NMR that is there, of course. Then there's something also very interesting about this database in that we have shown also a prediction of all the proteins that these compounds can potentially bind to. So in essence, protein compound predictions are all there. So in essence, when you find a compound of interest, you can actually want to know which of the proteins are possibly going to be the targets for these. So it gives you all the predictions for protein compound binding, and also ranks them up so that you can now see which of them, which direction you can actually go into drug discovery. So that already is a very useful platform for someone who wants to do drug discovery beginning from natural products. Beyond that, there is a complete annotation of every single compound in terms of the compound smiles, the compound name, the PubChem ID if they are in PubChem, the species where they are found, the traditional use of the species, the part that was used in the study, which means that it's possible to reproduce the study if you want to. For example, it tells you exactly which part of the species was investigated, if the sample is available in a herbarium, what is the number, and even the compounds in there, the compound classes and the subclasses, and any reported activity, the country, and GPS coordinates where the species was harvested for some cases, even the date of the harvest, because often the day the season of harvest will determine the quantity of the metabolites present in a particular sample. So all of this information, the authors and so on, we have all of that catalog in there. So it's quite a huge repository. Now, this for us could be the starting point. Of course, we are constantly working to update and to improve the quality of the database. But in terms of numbers, the information currently present could be is information published from 1961 to 2024. So right now, we just built a tool that will enhance the manual curation of data, which we hope to publish soon. And once this tool is good up and running, then we can update all the data that was published until 2026, until the date of today. Now, beyond that, what you observe is that most of the activities reported in the literature, the majority are cytotoxic, like the cytotoxicity represents the highest percentage. That would be something around 6%, while the rest will be quite varied. And then in terms of the source of the species, most of them are plants. While we have a few compounds coming from animals, mostly sponges, from marine sponges, and also a few compounds coming from bacteria and fungi. Now, if we go to the families of the source species, the majority of the compounds come from the Asteraceae, while we have quite a significant number coming from the Leguminosae and the Euphobiaceae. But the majority of the compounds are terpenoids, which of course we expected that because The terpenoids is the largest family of secondary metabolites in plant species. Now, when you go to a compound card, you will quickly see information there about the name of the compound, the structure, the family where the compound comes from, and the species, and so on, the classes and biological activities. So a lot is available. Of course, you can now browse through. So in essence, you can browse through by keywords, you can browse through by list of compounds, you can browse through by family, by species, you can browse through by literature references, and so on and so forth. But as I said before, more than 11,000 compounds present from 6 kingdoms, 264 families, of more than 1,700 species, and more than 191 recorded types of biological activities, and more than 2,000 references from the literature. And of course, about 9,000 of these compounds are annotated to PubChem. So which means if you want more information about these compounds, you can just go there. Now, beyond that, we try to ask ourselves what percentage of the compounds in ANPDB are found in coconut, for example. Coconut is a collection of 63 natural product compound databases, and there coconut has almost 700,000 compounds in there. And then we try to compare what chemical space is covered by our natural product library when compared to coconut. I would say that, first of all, coconut, when they were constructing coconut, we donated our data, the data that was available at the time from all our collection of library collections that we had made. So coconut has already, and we're expecting that we're going to have some superposition. So we have quite a significant superposition between coconut and our library. So about 5,400 compounds are found in coconut. But beyond that, we still have a significant number of compounds that are not there, that are not yet in coconut. Of course, hopefully in the next version of coconut, you're going to have all of our library there present. And there was this second nice story I told you, I always tell, is that with the use of computers, particularly if you have to run things like substructural searches and similarity searches, there is this principle, the similarity principle, that simply says that compounds with similar structures are most likely going to bind to similar targets. And at the time we just rounded up the first online version of our database, that was the NAN-PDB, that was the Northern African Natural Product Database. Our interest was to say which compounds in there are very similar to known drugs. We said, okay, let's try. So we tried to find which compounds are similar to artemisinin, for example, well-known anti-malaria drug. And we realized that ostracine is quite similar. Actually, the similarity index is the Tanimoto coefficient comparison of these two structures was like 0.7. And we found out that this compound was found in a species called Ambrosia maritima, which has been used traditionally to treat bilharzia or schistosomiasis. So we realized, hey, these two compounds look so similar, but at the same time, the species from which this one we didn't know before was identified has some anti-parasitic properties somehow. So in essence, we think that similarity search could be one of the potential ways of investigating or making use of the database that we have. There are many other case studies I will narrate here, and one of them is this indole here. And this indole, what's intriguing about this indole is that it was found initially from a species that is used to treat stomach ulcers, stomach disorders and fevers. Now, so visual screening led to the identification of this compound as having a potential anti-cancer property. And of course, the species have been used over the years just for treating disorders. No one knows what is behind it. So this could be nice to further investigate. Another thing we saw also is this arginine-based alkaloids, and these compounds that we call suberimines A and B, they actually were found initially from a sponge, and visual screening showed that these compounds were able to hold the entry of SARS-CoV into the host cells. And beyond that, the compound were also synthesized recently and tested and found that they have in vitro activity against several cancer cell lines. And there are several success stories in terms of visual screening from ANPDB, and one of them, of course, being this flavonoid glucoside here, which is seen to predict, which is predicted to block the entry of SARS-CoV-2 by binding to this NSP 10 to 15 complex, which of course is crucial for the virus invading the host immune system. But beyond just the natural products, how about the pseudo-natural products? Now, pseudo-natural products are defined as compounds which are not natural, we don't have a biosynthetic pathway for making them, but whose fragments come from nature. So I would give a quick example. Here's grismonon, and you see grismonon has two fragments. One of the fragments comes from gonitolide C, which has antifungal, anti-cancer properties, anti-diabetic properties, and the other functionality, other fragment comes from an antifungal metabolite. Now, when you fuse these two together, you form a hybrid. What you find is a compound that has completely different activity from the other two. So it has been shown that compounds, fragments from nature could make compounds that completely have different activities. So this is a field of research that we started investigating. And what do we do exactly is that we just collected, we just made collections of natural products in-house, and we had about more than 10,000 compounds which are natural. And what we did was that we performed a kind of retrosynthetic decomposition of our compounds, that what are the fragments that would make them if we were to synthesize them. And so we break them down into smaller structures and then filter them out. So we broke them down into the smaller structures, knowing that there's going to be redundancy. We had about 5,000 fragments, which we now apply the rule of three. The rule of three is simply a simple filter rule that determines which compounds could be lead-like structures, small molecule structures. Then these ones were about 800 of them. So these 800 fragments, what do we do with them? We had a target of interest, and this target of interest here was a human histone deacetylase 1, because it has some implication in the reversal of HIV in host infected cells. So dormant cells could be reactivated by inhibition of infected HIV cells, could be reactivated, which are latent, could be reactivated by the inhibition of the histone acetylase 1. And here is the protein structure of our target, which we got from the PDB. And what we did was we took all our fragments that we generated from the natural product library in Cameroon, in Africa, and tried to interact all of them in the protein binding site. And we scored all of the fragments. And then the top scoring fragments are the ones that we were finding interest in. So we tried to overlay all the fragments in the binding site and try to now see in which ways can we combine these fragments within the binding site to form a new set of compounds that were not in the library at the start of our deeper binding site. So when we now start overlaying each fragment at a time, the top scoring fragments, we realized that it was possible to actually combine the fragments to form molecules. And this combination it can only be feasible with the computer. So sometimes we'll determine if we have to use a linker, and then what with length of the linker, we vary several lengths, and then we try to dock them again and see if this fits well in the pocket. And then, so in summary, what we did was we did a library collection from Africa. Several at the time, NPDB was not yet completed, so we just took all the libraries available. We did a retrosynthetic breakdown of the fragments, and then of course we filtered out for pains and toxicity alerts and so on, things like that. We removed those compounds that can potentially interfere in the assays. Then we applied the rule of three. We docked all the fragments and we tried to recombine them again. And then in the course of that, we selected a couple of scaffolds. And among them, we selected 3 scaffolds. One of them, of course, was indole chalcons, which we have already published. And in total, we made about 51 pseudo-natural compounds. 51 of them were synthesized, and they were assayed in the JLAD assay, which is able to identify from there the compounds that could have the reversal of the dormant HIV. Then of course, from there, we found one active compound, which I cannot show you the structure because the patent has not been approved. And then we have this scaffold. And what is interesting about this scaffold is that this particular compound, which was actually unique compared to the other 51, had some inhibition of it was able to reverse, some HIV latency reversal properties, and also inhibited human HDAC1. And what's interesting is that it's HIV latency reversal property similar to that of prostatin, which was the standard HIV latency reversal agent. And another thing we observed is that, of course, as you see, most of the compounds were either inactive completely or moderately active. Just this one stood out. And when we went to dock the compounds, the active ones and the moderately active and inactives, we quickly realized that the docking score of the active was quite distinct compared to the moderately active and the inactive. We also observed that where we decompose because we use G-score, which is an empirical scoring function that is able to decompose the score into several contributions. We realized that the highest contribution was the van der Waals contribution. So in essence, that was expected because the binding site is quite hydrophobic, and we realized that the van der Waals contribution is the highest contribution towards binding to the protein binding site. And the work, the first set of indoor chalcones that was published is described here, while the remaining 40 compounds are also shown in this paper. That's almost out there. And once the patent is approved, then we will get to approve the publication of that paper. And so beyond that, we are sitting on a continent with a lot of effort going on. So we think it's important to concert, to put to bring our own contribution towards drug discovery. And one of the activities we are involved in is in this African Grand Challenges Africa Drug Discovery Accelerator. And in this consortium, there are several groups, and in principle, there are 4 flagship projects. Ours is just one of them. So in this consortium, our work is to set up a box of natural compounds, approximately 400 compounds, which you can make in several copies and get them screened in several biological assays. The idea is to map the activity landscape of natural compounds coming from Africa. And we also have the group of Professor Richard Amewu, who is in Ghana, actually leading the drug innovation group and also leading the malaria flagship project in GC ADDA. We also have Professor Lynn Marie and Eric Strauss in Stellenbosch, who are both involved. Lynn Marie is involved in the malaria project, while Eric is involved in the TB project. We also have Professor Colin Massimirembwa, who is leading the DMPK Center of Excellence in Harare. So all these efforts come together, pointing towards proving that actually it's possible if we overcome some of the challenges we have. Drug discovery in Africa, it is a possibility. And so this calls for a collaboration. And I will say quickly that there are a couple of things going on, and we really need collaboration in the African derived natural product box. So you can always reach out to me and see if you are able to make contributions in this project. And if you have a new compound, or you have a compound which is of interest, but you don't know in which assay this compound could be possible, of course, you can reach out to me. We'll see what are the possibilities of including this compound in the box. Another area of call for collaboration is that we are building in vitro screen platforms as well as validation assays. So if you have some skills, or you are interested in getting skills in vitro screening, we are quite there, willing to work. We are hosting an intern from Nigeria, and next month we are receiving an intern from France. So we are quite interested to collaborate. And when I look at the future, what I see in the next 5 to 10 years is my focus right now is capacity building, trying to pass on my skills and my knowledge to the next generation. And very interesting for me is the fact that AI and machine learning can fast track the discovery of bioactive compounds. So one of my PhD students shown here, Jude, is just rounding up his PhD, and he has found quite a number of SARS-CoV-2 compounds, and some of them have already shown activity in the assay, and all what he was using was generative modeling and visual screening. And I would also say that to save the African plants, to save the species. It's also interesting that we, beyond just building an African pharmacopoeia, which one of the efforts I'm spearheading at the continental level, we think it's important that species of interest, which are at risk of being of extinction, could be sequenced, and the genomic data of these species kept in biobanks that in future can be exploitable. And also, species, instead of going all the time to the forest, it's now time to start using more advanced techniques, including expressing the genes of bioactive plants in heterologous organisms that could now be making the metabolites of interest, instead of all the time going back to the forest to kill our plants. And in conclusion, I would say that natural products in Africa is an untapped resource, and we know that this database we have built is a huge cornerstone for research and development on the continent, and that the use of artificial intelligence could fast track the discovery of this. And we have several pipelines that we are building together with Esilia, and we know for sure that inspiration from nature could drive global health innovations, including finding new therapeutics for some of the diseases that affect the continent. But of course, for that to happen, partnerships and collaborations are a key to that success. And I will not end without saying that this would not have been possible without funding from several partners, including the support we are getting through H3D, through the Grand Challenges African Drug Discovery Accelerator Program. And of course, the Gates Foundation has been there to give us the push, as well as LifeArc. And I will show you a couple of pictures of our labs. We didn't show the HPS we just set up recently, but we have a couple of pictures to show the facility where we work, and what our typical day will look like. A typical day, some people will be screening compounds in the bioassays, while some will simply be trying to filter out compounds that are from libraries by interacting with proteins. And a typical day, we would say sometimes we have workshops, and the picture here is looking like 2 years old. And this was a workshop we had here together with Ian and Mikel and Gemma. We had hands-on workshop with colleagues coming from Tanzania, some coming from Chad, some coming from other African countries. And so I would say this effort would not be possible without the dedicated team here. We have various labs, our teams in computational chemistry, natural products, data entry team, administrative team, and synthesis and biological assay. So, and of course, they say in Africa that what a young man sits down, is when an old man sits down doing, a young man climbs on the tree doesn't see. So we also benefit from the consultancy of very experienced professors here. And of course, we are quite willing to take your questions. Please reach out to us if you are interested in collaborating. You can send us an email. You can visit our website. You can go to the African Natural Product Database website. Of course, you can reach out to Amazon and pick some of our books, and that'll be it. So thank you very much, and looking forward to your questions.
Fidele, thank you so much. I've unmuted everyone, or enabled that function. So feel free to raise your hand if you'd like to line up to ask some questions. But we've already got one in the chat, in the Q&A section from Yamisi. Fidele, you alluded to this earlier. Are there instances where the compounds indeed work synergistically and no single isolated compound can reproduce the therapeutic effect?
Yeah, of course, there are many cases like that. We have cases where, of course, where an extract is more active than the isolated compounds. So we have some other cases where the authors will certainly will go further and try to see if the synergism observed among different extracts and compounds. So some of these ones are reported in the literature. But unfortunately, when we report in the A and PDB, we just write a comment to say that this compound synergizes with this, but we don't actually show, because we don't have the possibility of, we just show the individual compound activities and say there's some synergism with this other compound.
Thank you very much. Much appreciated.
Thank you, Yumisi. And then a question from Mohammed Mega. How many compounds from A and PDB are approved?
None. But there is there are a couple of teconolites which are already in clinical trials for treatment of cancer. Teca species, they actually teconolites. They're quite complex structures, and I would say these are the most. Of course, we know that Artemisia annua grows in Africa. There are some farms in Rwanda, I think, where there are huge population plantations where this grows. So where we find that these species like that have been investigated, we'll include it, of course, which means that Artemisia should be there, right? And so that should be some interest. But of course, that would not be news, of course. And what else? So we also have UMBIN, of course. UMBIN is there, and of course it has a very pronounced activity. And of course we know the reasons why UMBIN is not in clinical use now. But of course the species is being used by population. That's it. I don't know if that answers the question.
So Mohammed, if you'd like to unmute yourself, you're welcome to enter in discussion. But yes, I mean, this points to some of the challenges that you're facing in spearheading this work. In integrating such a large amount of data within more global systems. Okay, Mohammed says yes. All right, do we have any other questions? Feel free to raise your hand. Just want to see what's come up. If anything else has come up. Thank you for your presentation. Understanding the infrastructural constraints in sub-Saharan Africa. How do you achieve structural characterization and elucidation of isolated compounds from plant extracts?
Now, the question is twofold. Yes, the question is twofold. The first thing I would say is that there are compounds we collect in the literature, which we are not the ones responsible for the structural elucidation. So we take the structure the way it is in the literature. And if we doubt the structure, we can actually compare with structure that's published elsewhere. For example, we can go to ChemSpider and compare this. We can go to PubChem and verify this. We can go to Zinc, for example. We can just do a comparison. So in essence, when we have a structure in the literature that is published, we take it the way it is. Now, if for our own compound collection, like the physical compound collection, the African derived natural product box, there we use both mass spectra and NMR to elucidate the structures. And if you are really interested, we're actually planning a workshop on structural elucidation, which we have not yet decided. But when that happens, we are going to advertise that.
Okay, thank you. Then Bakari, go ahead and unmute yourself.
Hi everyone. Thank you, Nicola. Can you hear me first?
Yeah, I hear you, Nicola. I hear you, Bakari.
Hi, Prof. Thank you for the nice talk. So my question, you answered the first part just now. So my question is about the potential of discovery of new natural products in Africa. So I'm thinking like coconut is about almost 1 million now, and I'm seeing the natural African natural products going into the 10K. So there is like a huge disproportion in terms of natural product from Africa compared to all the natural products in the world. So what is I guess it should be like there are more products to be discovered. And also, how do we overcome the financial burden of structural elucidation? I think that's the main barrier now. And the last question would be about how do we uncover the traditional knowledge of traditional healers and traditional practitioners to come up with new, new sources of drug?
Now, the first thing I would say, Bakari, those are quite interesting questions. The first thing I would say is that coconut is making no effort. Between us, we know this because they're collecting from everywhere. That's it. We spend efforts going to the literature and mining literature manually. So it takes us a lot of time to put together 10,000, 11,000, 15,000, which if we collect the data that's already in-house that we have curated and the literature that we have not yet put in there, it could take us easily to 20,000. For us, we are quite happy because we are on our lane. So what Coconut is waiting for is that we publish these data, they just collect it and and put it in. So they just collect, they're piping from everywhere, even though of course they are now doing data mining themselves. But then if you talk about data mining they're doing, if it's blind data mining, because we doing as chemists, we do manual curation, so we are actually careful to verify that this information is correct. Yeah. So then I would say that it might look insignificant, but compared to the effort we're putting in, is quite enormous. And knowing that the data that we're generating locally from libraries and so on would not have been accessible to Coconut if we did not do that work, right? And so comparing, that's concerning the first question. I would say quickly that the data we have in-house, if we put all of that together, plus, because that was up to 2024, and not all the literature is even there. So there's some information that we are still cleaning up before we can upload. So that could take us easily to 20,000. So this is just like a template to work with. Then regarding the cost of investment in NMR and all that, yeah, it's quite enormous, right? And unfortunately, our governments have money to fund sports and other things, but they don't have money to fund science. So if our governments will fund efforts like this, we are going to be a lot more efficient. Now, I end my story there. And concerning the traditional knowledge, how to capture that information is quite challenging, because we are dealing with different communities. And in some communities, they are very skeptical. They're very skeptical because they think you come and get their knowledge and they'll get nothing in return. So if the Nagoya Protocol is carefully implemented in our communities, then of course this question of trust will not be there anymore. But of course, most of the traditional knowledge information is passed from, along the lineage. So somehow there's hope that information is in there. And of course, we have a lot of charlatans as well. So how do you verify the information and all that? So that's the challenge. Of course, we are right now in the middle of it all, and we cannot go backwards. Yeah, thank you. Does that satisfy you?
Okay, thank you, Prof. Yeah, that's it.
Okay.
Yeah. Thank you, Prof.
I also see.
Thank you.
Yeah.
Ayinde, yeah, we've got another lined up. Go for it.
Mm-hmm.
Thank you so much, Prof, for the exciting presentation. I just wanted to ask, in the course of your lecture, you mentioned that you have some mentorship program for young African scientists interested in drug discovery and development. So I don't know, how can we, for those that are interested, how can we apply for such mentorship program? And the second one is, do you have any group for publishing these findings from different research findings for drug discovery related research findings? Do you have any group that is focused on, the publication of these findings? If there's any such, I'm interested. Thank you.
Yeah, the first thing is that we host interns. We have limited capacity in terms of how many we can host. So, for example, our simulation lab I just showed you has 12 workstations, so we cannot take more than 12. So at every time we have students coming to learn basics of computer-aided drug design, virtual screening, AI, and all that. They do that in turns. So right now, the person from Nigeria will be going at the end of February. Then someone else is coming from France to spend, especially originally Cameroonian, will be spending 6 months with us, and so on. So in essence, the way we manage that kind of training will be like, you write a proposal and send to us what you want to do, and we will try to see where we can get funds to host you. And then of course, if you can self-fund, that'll be really ideal. If your group is self-funding, that's ideal. But if we must be the ones to host you and cover your living cost and your transportation, we will see if this fits within one of the projects we are doing, then that will be fine. If not, then we try to find out how to get the funds. So usually we handle it on a case-by-case basis. Now, concerning groups that publish, I don't really understand what that means, but I would say that you can see that we publish our work. So in essence, when we get results at the point that we think is interesting enough to publish, they will write the publication and publish. I don't know if you wanted to be part of publications or you wanted to collaborate. I don't know exactly what you mean.
Yeah, I actually mean collaboration, because I'm a pharmacist. I'm a recent graduate, and I've actually published a number of papers on computer-aided drug discovery. So,
Yeah.
and I'm willing to collaborate with other researchers from Africa. So that's why I actually put out the question.
Okay. All right. All right. Then, of course, I think I left my, if I'm not mistaken, my last slide is still on the wall, so you can actually get my email, and we can talk a lot more detailed after this talk.
Thank you, Fidele, for being so available. I'm also popping some links in the chat as well, which might be of interest to you and others. We've got time for just one more short question. We've got a hard stop on the hour. Jamanze, is it Jamanze? Go for it.
Yes, it's Gamanza. Greetings to everyone. Thank you so much, Prof, for the presentation. Yeah, we are short on the hour, but I have one comment and one quick question. The comment is that I'm a master's student in antimicrobial resistance, and I'm currently doing a virtual screening of natural compounds targeting the multi-drug efflux pump of Klebsiella pneumoniae.
Okay.
And ANPDB is one of the databases I am using. And I want to seize the opportunity and thank you, Prof, and the team for the good work you are doing, because you work on these databases, you put the compounds in electronic format, so researchers around the world can use it for free. So I'm very grateful for that. And my question is, during the presentation, you spoke about combining fragments, and this combination yielded different activities. Now I'm thinking about the possibility. What about combining these natural product fragments and also fragments from existing drugs, if it's possible, and if it could yield potentially another field of research? And as well, I'm interested in the intended opportunities, and I heard what you replied for the previous person, so I may get to you through the email. Thank you.
Okay. Yeah, I like the idea. So far, what we have done is we have combined natural product fragments. So it doesn't stop you from, for example, generating fragments from drug bank and try to combine fragments from drug bank with fragments from NPDB. It doesn't stop you from. I would typically say the easiest way, if you want to do fragment-based design, is first of all to be sure that you have access to the fragments. That means we are screening a fragment library. You should screen a fragment library that you can, first of all, assay the fragments themselves. And if the fragments already have some activity, then it will be possible now to say, if I combine the activity, if I combine my fragments, it may enhance the activity. It's just a principle of additivity. That means that if one fragment is interacting with one portion of my biocide, then the other fragment is interacting with the other portion. If I combine the two of them, most likely the activity increases, right? So I would say ideally, if you have, I can show you several fragment-based fragment libraries that are available on sale. That you can actually screen them virtually, and then when you have some hits, you actually order the fragments and then assay the fragments before now combining them to make compounds that you gain assay. I would say that would be the best approach, because of course, when you are screening NPD, where you're done, I know you're going to send me an email to ask me where we get the compounds from, and I'll give you the answer that we don't have the compounds. So in essence, we have a few, but we cannot just give all of it. So I would point you to Pineapple Library and other things. But anyway, we are open, and I look forward to getting your email.
Okay, Prof. Thank you so much for
Welcome.
the feedback, for your reply.
That brings us to the end of this week's webinar. So it leaves it to me to thank all our participants for joining us. We host these webinars on a sort of ad hoc basis, but we try to get 2 in a month. So please look out for the next one. I've populated the chat with various links, including a feedback form if you'd like to suggest future speakers or give us your feedback on any of the topics and speakers that we're presenting at the moment. So a huge thank you to you for joining us. And then, of course, to Fidele for sharing your knowledge, passion, expertise, and professionalism. We are so grateful and so excited about what you're doing on the continent, and pleased that we can, play a small role in it and support what you're doing. Thank you so much for giving your time today. We so appreciate it.
Welcome. Bye.
Bye, everybody. Until next time. Thanks again.
Bye.